Hi, everyone. The video you are about to watch is part of the Age Presents lecture series. The series is produced by the American Aging Association to provide an introduction to the foundational and advanced topics in geroscience. As you watch this video, I would like for you to keep 3 points in mind that will help you understand the importance of biological aging. 1, aging is the primary risk factor for disease, including cancer, metabolic syndrome, and most neurodegenerative diseases. 2, the biological approaches that you will learn here today, such as model systems that help researchers learn more and understand about these types of diseases. And 3, mostly deals around you. What is your interest in aging research? Whether it's biological, translational, or clinical, how would you like to contribute to the field? And if you're not already a member of the American Aging Association, I would encourage you to visit our website to learn more about us and consider joining today. AGE members receive free access to the Society's journal Geroscience, reduced registration for our annual meeting, opportunities to interact with many of the leading scientists within the field, and access to these lectures, slides, and notes used here today. And there's more. We have a very active trainee chapter with many opportunities for networking and career development. And last but not least, I would like to thank our sponsors for the Age Presents lecture series. The University of Washington Healthy Aging and Longevity Research Institute, the Jackson Laboratory Aging Center, and the Nathan Shop Centers of Excellence at both of these institutions. Thanks for listening and enjoy the lecture.
Okay, hello, my name is Holly Van Remmen, and I'm professor and chair at the Oklahoma Medical Research Foundation. And I've been working on oxidative stress and aging for the great majority of my career. And so they've asked me to put together a lecture today to go over some of the basics of this area for you. So we all know that aging is associated with a general loss of homeostasis and a deterioration in a number of structural components in our bodies and in our cells, and a loss of cellular maintenance, as well as a decline overall in just general physiologic function and metabolism. And together, all of these lead to an increase in the incidence of disease as we age. And so what we don't know, though, is really what causes aging. We still don't know. And when I came into this field as a graduate student, I would say, which was many years ago, I would say that the theories of aging were basically broken into 2 different groups. There were a number of theories that focused on programmed effects, so time-driven events in response to different changes in gene expression. So the idea was aging programmed? I mean, is it something we can't really alter? It's just a programmed effect. And the other set of theories were focused more on damage, so the accumulation over time of insults that we all receive from the environment and from internal sources of damage. And so one of these more prominent theories at that time was the free radical theory of aging, which was first proposed by Denham Harmon in the 1950s. And he was actually working on radiation chemistry. And it occurred to him that there were changes that were a part of normal metabolism that as a result of mitochondrial function or other sources in metabolism that would cause the formation of what are known as free radicals. So these are damaging species in the cell that then can cause damage and over time lead to aging and age-related disease. This was thought to be as said by him, a single common process that could be both modified then by genetics or by the environment. And this would be then responsible for the death of all living things. This is I think, a very satisfying theory in that where he was just proposing that normal metabolism and changes in the generation of these normal metabolic byproducts called free radicals could then lead over time to all of the different phenotypes that we see as aging. So that brings up the question of what are free radicals? So free radicals are molecular species that contain an unpaired electron. So that makes them rather unstable, and they can cause all sorts of problems, right? Damage to other molecules. And the way one way we can combat these is with an antioxidant that provides back the electrons and then makes everything stable again. So these free radicals can have effects throughout different components in the cell, to DNA, to protein, to the lipids in the cell, and cause damage. So the next question is, well, where do these free radicals actually come from? And the primary source of free radicals is generally thought to be the mitochondria. So in fact, about 90% of the free radicals that we see come from the mitochondria. And this comes as a result of the process called oxidative phosphorylation, which helps to generate ATP and energy source for the cell. So this is a primary function of the mitochondria. And so as they're moving electrons down along the electron transport chain, which is present in the inner membrane of the mitochondria, some of these electrons can leak and react with oxygen and cause the formation of free radicals. In particular, as shown here, oxygen can be converted to superoxide anion, a very harmful free radical that can then be also converted into hydrogen peroxide, which is a very freely diffusible cell. And you can see diffusible species, which you can see here by the arrow, can actually then move out into outside of the mitochondria into the cytosol and cause damage to other parts of the cell. So it's generally thought that about 1 to 4% of all the oxygen that's coming into the mitochondria going into oxidative phosphorylation to generate ATP, about somewhere between 1 to 4% of this oxygen can be converted to reactive oxygen species. And these estimates have changed over the years, but I think in any case, it's a substantial amount. So in addition to the non-mitochondrial sources of free radicals, we also have a number of external sources. So for example, there's radiation and UV rays that are out in our environment, and there is pollution that can come from a number of sources. And within the cells, there's also a number of other non-mitochondrial sources, including enzymes such as NADPH oxidases, cytochrome P450s, cyclooxygenases, and a number of other processes. In fact, one significant one is the production of superoxide by phagocytes. So we need to remember, though, that while mitochondria are generating these reactive oxygen species, which is thought to be, a deleterious aspect of their function, mitochondria actually have a lot of very important roles in the cell. And so here's a schematic of a mitochondria showing the electron transport chain. But we know that one of the other things mitochondria do is they're responsible for initiation of apoptosis. And this is important because there are times when we need to have apoptosis and cell death. And this is initiated by the mitochondria. They also, by producing these reactive oxygen species shown here as superoxide and hydrogen peroxide, they also contribute to ROS signaling, which is not necessarily a damaging effect, but actually puts into effect many cell survival pathways as well. Of course, there's ATP production, as we talked about. Mitochondria are also very important for calcium regulation, which is critical for the cell, and other processes such as fatty acid oxidation. So the mitochondria are very important. They don't just have a downside. So the bad side, though, of course, is the fact that these ROS can cause damage. And as we mentioned earlier, this can occur to different components of the cell. And so more specifically, if we have oxidative damage to lipids, we can get changes in membrane peroxidation, changes in the fluidity of cellular membranes, which can greatly affect the way that the membranes are working. And the marker that we use generally to follow lipid peroxidation is the F2 isoprostane. So this is a lipid peroxidation product that can be measured very sensitively and accurately by GCMS. And then we can also get oxidative damage to proteins. So sulfhydryl groups in proteins can be oxidized. They can lead to reactions with aldehydes. And very importantly, this oxidative damage can often lead to protein aggregation, which we know in particular neurodegenerative disease has had a very deleterious role. And so here we look at protein carbonyls. And then finally, we can also measure oxidative damage to DNA. In this case, the damage to the DNA can lead to mutations and deletions. So this is obviously very important for things like cancer, but these changes in the DNA can be implicated in a number of different processes. And here we use an HPLC-based method to measure 8-oxo-DG mutation in the DNA. And then, as we said, on the good side, the mitochondria can do many beneficial functions, including, as we said, changes in this ROS signaling pathways, playing a role in cell proliferation, and many other things. So it's really a balance then between the good side of the mitochondria and the bad side of the mitochondria. So our cells have developed a very intricate network and a system to control this generation of reactive oxygen species. These ROS are not just allowed to go rampant and cause damage and have all deleterious effects. So there's a very complicated system of antioxidant enzymes that are present in different parts of the cell. So here I'm showing in blue the mitochondria, and here we have enzymes such as manganese SOD that can scavenge the superoxide anion and convert it to the hydrogen peroxide, copper zinc SOD, which also scavenges superoxide. And then in the cytosol of the cell, there are also very important antioxidant enzymes that can scavenge these species that are released from the mitochondria. So these enzymes have very specific targets, as shown here on the right. The SODs looking at superoxide, glutathione peroxidase and catalase that target the hydrogen peroxide, and there's enzymes for the lipid peroxides and for protein damage as well. So there is a very elaborate system for protection and detoxification. So in addition to these enzymes, though, there's also a very important contribution of redox homeostasis. So the redox couples NADH, NAD, NADPH, NADP, and changes in the ratio of oxidized to reduced glutathione. All of these are part of what's called the redox network, and they also play a very important role. So it isn't just about these primary antioxidant enzymes, but there's a very complex system that works together to control oxidative stress. And I think that's a very important point. So the free radical theory of aging was eventually thought to not really completely capture what was going on, because it's not really all about this for the free radical generation, right? It's also about this idea that there are compensatory mechanisms there. So it was thought to be more accurate to talk about oxidative stress. So this is a little bit of a different concept. So we still have these sources of reactive oxygen species, and we have antioxidant defense mechanisms to balance them out. And then what we end up with is sort of a net result. So oxidative stress then is when we have an imbalance between our capacity to be able to defend against the radicals and the actual production of the radicals. And so if this imbalance persists, you can have a chronic state of oxidative stress. And then that in turn is where we get the damage to the cellular components. And over time, we have an increase in oxidative damage with age and a progressive loss in the efficiency of different cellular processes. So Dr. Raj Sohal actually first proposed this modification of Denham-Harman's theory in this paper in 1990. And I think it's really a better way for us to think about it. And Dr. Sohal and his colleague, William Orr, actually did some pioneering work in this area of the importance of oxidative stress in aging. And in this case, they were using Drosophila, flies that had been genetically manipulated to overexpress superoxide dismutase, which we said can scavenge superoxide, and catalase, which can scavenge hydrogen peroxide. And so the important part is that they generated flies that overexpress both of these enzymes. So that way you can remove the superoxide, but then the hydrogen peroxide that you produce will also be removed by the catalase. So this was a sort of a tandem effect. And you can see in the panels at the bottom that when they overexpressed both catalase and SOD, they had, if you look at the mortality rate, you can see that in the control, for example, the median lifespan was 54 days, but it was much higher in the flies that had the overexpression. And this is illustrated in the diagram here at the right. You can see lines A, B, and C that live longer than the control strain. And importantly, they asked the question of, well, what does overexpressing these enzymes actually do with respect to oxidative damage? And you can see the upper line is the control, while these three lower lines are in the overexpressing flies. And you can see that protein carbonyl, protein oxidative damage was actually dampened in these animals. So this was really a key study that lent sort of the first really strong support to the importance of oxidative stress in aging. And so about not too long after Harman put out his original free radical theory, he also sort of backed off a little bit and said, or modified the theory just a bit to bring into play the importance that the mitochondria were playing in this whole thing, because we had said, well, mitochondria are the major source of ROS. And so he actually thought that the mitochondria might be a large key to this free radical theory. And he also said that the mitochondria, by producing these ROS, could actually produce free radicals that would actually affect directly back at the level of the mitochondria. And so then in this case, you could get damage to the mitochondria, which might cause a feed-forward effect for further production of ROS. And so, you know, just a reminder, we have this continual generation of ROS in the mitochondria at the electron transport chain. We have substrates feeding into the electron transport chain, electrons going in, and we have the electron leak that causes the generation of these free radicals. And primarily, this leak is thought to occur at complex 1 into the matrix, or at complex 3, both into the matrix inside the mitochondria, as well as into the intramembrane space where the radicals can easily get access to the cytosol. So again, this mitochondrial theory of aging was proposing that this, electron transport chain, ROS activity would, damage the electron transport chain itself, and that would lend to further production of ROS. And again, over time, you would get more and more damaged mitochondria, which would reduce your ability to produce ATP, alter changes in apoptosis, many of those other beneficial pathways that we talked about earlier. And in general, this mitochondrial damage would then lead to aging and age-related disease. So that's sort of the background. And what I wanted to do today is to give you 3 examples of studies from mouse models that sort of interrogate whether or not this theory makes sense. And so the first story I'm going to tell you about is about manganese SOD, which is the mitochondrial superoxide anion scavenger. And so there was a study done a number of years ago now, where they generated a mouse model that was deficient in manganese SOD. So as you can imagine, if you don't have enough manganese SOD in the mitochondria, you can't scavenge the superoxide efficiently. And so superoxide anion will then build up. And this study came out of the laboratory of Dr. Charles Epstein at UCSF. And these mice were very interesting because when they completely removed manganese SOD, what they saw is that the pups that were completely lacking this enzyme were much smaller, they were paler, and had severe phenotypes both to cardiac and to brain tissue. And in fact, in some strains of mice, the null mutants weren't even born at all. But even if they are, they only would last for less than 2 weeks. So the complete lack of a manganese SOD is not consistent with life. So we know already that it's playing a very important role. This was actually a project that I worked on in my postdoc, when that was the idea to then take the heterozygous mice that we know were viable and see what the effect might be on aging and age-related pathology. So the first thing we did is to take the mice and look at the levels of manganese SOD in wild type and knockout mice, and you can see, as you would expect in a heterozygous knockout, with 50% of the genes present, so we have about 50% activity in a number of tissues. And so the most important question to us, though, was, so what happens to mitochondrial function? And so we looked at mitochondrial ROS generation indirectly in this case by looking at the activity of an enzyme called catalase, which is present in the mitochondrial matrix, and it's known to be sensitive to inactivation by superoxide. So if there's more superoxide, you would expect the activity to be decreased, and that is exactly what we saw. And in parallel, we also looked at another enzyme, NADH oxidative reductase, which is also sensitive to oxidative inactivation. And again, you can see there was a decrease in activity. So these two activities suggested that there definitely were higher levels of superoxide around in response to less SOD2. And when we looked at the actual function of the electron transport chain complexes, as far as their ability to consume oxygen, we measured the respiratory control ratio. You can see here that in all 3 primary electron transport chain complexes, there was a decrease in activity. So together, this all suggested that this loss of SOD2 had an effect on mitochondrial function. So the idea here was that we wanted to test the mitochondrial theory of aging, right? So if the theory is correct, and if we have this model where there's a lot of increased ROS production, then that should in fact cause oxidative damage and changes in mitochondrial function itself. And potentially this could also increase the sensitivity to further oxidative stress. So if we were to use exogenous generators of superoxide, we should definitely see a reduced capacity to protect from this. And that at the end of the day, these should accelerate aging. So we had seen, in fact, that we're seeing increased ROS, increased mitochondrial dysfunction. So now the question was, what happened to damage? So we looked at DNA oxidative damage, and you can see here in blue versus yellow is just the effect of age. So many people have previously reported that oxidative damage increases with age, and you can see that in both the wild type and the knockout, in general, we saw a difference between the young and the old. But what is striking here is that if you look at the values for nuclear DNA oxidative damage in the knockout, it's always higher than it is in the wild type animals. So there was an increase with age, and it was higher in the knockout, which is all consistent with the mitochondrial theory. So interestingly, though, while we saw major changes in oxidative damage to DNA, we didn't really see much change in protein carbonyls, so protein oxidation, or plasma F2 isoprostanes, which are, again, the lipid mediator, although there was a tendency for there to be an increase with age. So we also looked at the sensitivity of these to paraquat toxicity. So as I mentioned, this is an exogenous compound that you can give to the mice that generates excess superoxide. So you can see in young wild type animals, if we inject them with paraquat and look over the next 10 days, nothing happens with respect to their viability. However, the young knockouts, there was an increase in loss of survival in response to this insult. And if we looked at the effect of age, you can see here in the red dotted line, the old mice were much more sensitive than the young wild type mice. And the old knockout mice were the most sensitive. So again, this is consistent with an increase in sensitivity in these animals. So the real surprising finding in this study was the fact that when we looked at the actual survival of the two groups of animals, so the wild type and the SOD2 heterozygous knockout, you can see here that there's absolutely no difference. So, you know, we fully expected, if the mitochondrial theory were true, that you know, the increased ROS, increased damage, increased sensitivity would have translated to a change in lifespan, but that's not what we saw. So we also were fortunate at the time to have access to pathology analysis. It was done by Yuji Aquino in San Antonio. And what Yuji found is that if he looked at the old mice, wild type versus knockout mice, even though there wasn't a change in survival, he saw about a 3-fold increase in the incidence of lymphoma. Now, lymphoma is typically the cancer that mice are prone to getting, that kills them. And there were a number of other pathologies, but no real difference between the groups. But this 3-fold increase in lymphoma was really significant. And also, if you looked at the tumor burden, so the number of different types of tumors that the mice had, the incidence, the number of mice, percentage of mice that had tumors, those were also elevated in the knockout group. Although once the mice had tumors, there was really no difference in the severity between the wild type and the knockout. So, you know, the bottom line on the story for the SOD2 mice, as I said, is that everything was consistent with the mitochondrial theory as far as, you know, increased ROS, damage, changes in mitochondrial function, but yet the survival was not changed. So this really is not consistent with either the oxidative stress theory or the mitochondrial stress theory. Now, I mean, I have to add the caveat that, you know, as I mentioned early on, the antioxidant defense system is very complex. you know, it's probably not correct to make have any conclusions based on just manipulating just one antioxidant enzyme. You know, there's many other changes in redox that we're not probably catching here, that we don't really know the whole story yet. It still seems that you know, this is a really important finding, that that maybe Harman's theory, as initially proposed in its simplest form, is maybe not explaining the whole story. And you know, this even goes further, because we, after we found the results in the SOD2 knockouts, we looked at a number of other mouse models where we removed antioxidant defense. So you can see a number of major players here: manganese SOD, copper, zinc SOD, GPXs, thioredoxin, methionine sulfoxide reductase. And of all of these, the only one that showed any change is the copper zinc SOD knockout. But the rest all look, the lifespans are completely similar. It turns out that this is actually a really interesting model that I don't have time to tell you about today, but it also has a number of, an acceleration in a number of age-related phenotypes. So when we saw these results, we said, well, maybe, you know, just knocking out the one is not really enough to shift the system. So we did a number of crosses of more than one knockout. So for example, you can see here, if we knock out both manganese SOD and glutathione peroxidase, which is a hydrogen peroxide scavenger, we still didn't see any change in lifespan. So we looked at the flip side. So instead of trying to accelerate the decrease in survival, what happens if we try to protect the mice from oxidative damage? So we generated a number of different models with elevated antioxidant protection, shown here: copper zinc SOD, catalase, SOD2, glutathione peroxidase. And you can see, again, the story was very consistent in that manipulating these antioxidant enzymes does not affect lifespan. And in fact, again, if we use double transgenics in tandem, we still don't see any protection. And, you know, this was also not just from our group, but Dr. Charles Epstein, who had generated the manganese SOD knockout mice, also did a lifespan study before ours, actually, in the copper zinc SOD transgenic overexpressing mice. And you can see he was able to get about a 2 to 5-fold increase in copper zinc SOD expression, but yet no change in lifespan. And I can say that this was actually really very difficult for the field to sort of get their heads around, because I think, you know, I think I mentioned earlier that the oxidative stress theory is really a very satisfying idea, right? Increased damage over time, this could affect aging and lifespan. And so these results actually were a little bit difficult to understand. So we'll just leave that there for now, and we'll move on to another example. So I want to talk a little bit about the mitochondrial DNA polymerase mutant mice. So this is a little bit more of a direct test at the effect of the mitochondria. So there were actually 2 different models that were generated very close to the same time that sought to manipulate the proofreading enzyme inside the mitochondria, DNA polymerase gamma, which is, you know, important for maintaining the integrity of the mitochondrial DNA. So the idea here is that if you can maintain the mitochondrial DNA, you should be able to affect lifespan in a positive way. And conversely, if these mice that had a lower ability to perform this proofreading mechanism, you would expect them to live shorter. So this first group that was headed by Alexandra Trifunovic, showed that there was about a 3 to 5-fold increase in point mutations and deletions in the mitochondrial DNA, and that the mice looked pretty normal until about 6 months or so, and then they started to lose weight and to accumulate a number of accelerated aging phenotypes. And as far as the mitochondria, these point mutations, deletions were also associated with a reduction in electron transport chain activity and the production of ATP in the heart. So in this case, the elevation in mitochondrial DNA mutations was definitely associated with a decreased lifespan. So you can see here that these mice were very short-lived and had died with by 48 weeks. The median lifespan was approximately 48 weeks, or just less than a year. So this was pretty strong evidence that you could definitely alter lifespan with changes in mitochondrial DNA mutations. And there was, as I said, there was a second model that came out at the University of Wisconsin pretty soon after with a targeted mutation of polymerase gamma. And again, affecting the proofreading ability. And this study is really important because it sort of also ties together mitochondrial function and oxidative damage. So they definitely saw the accumulation of the mutations. They saw these same sort of phenotypes of accelerated aging. But it was interesting because these changes in the mutations did not have any effect on markers of oxidative stress. So this suggested that you could have deficiencies in mitochondrial DNA that don't necessarily convert to increases in oxidative stress. And so again, these mice look pretty normal in the beginning, but within a few months they develop a number of age-related phenotypes, weight loss, hearing loss, loss of muscle mass examples, as examples. So, the question here is, so do these studies support the mitochondrial theory of aging? I think it's not black and white. It's not cut and dry because, it's really hard to say what the effect of these mitochondrial DNA alterations are. So maybe they are just really, further advanced than anything that we would normally see in aging. So it's something to think about, but it definitely does suggest that the mitochondria have play an important role in a number of age-related phenotypes. And I think this question of whether DNA mutations, mitochondrial DNA mutations, are important or critical for aging is still kind of lurking around. I don't think we've really completely resolved this question. So then finally, we move on to the third story, which is a mouse model that was generated at the University of Washington in Seattle, where the expression of catalase, which is a hydrogen peroxide scavenger, was targeted to the mitochondria. And this is really interesting because catalase is not normally found inside the mitochondria. It's normally a cytosolic enzyme. But they were able to genetically target catalase expression to the mitochondria with the idea of decreasing oxidative stress. So this paper was published in 2005 in Science. And again, so what they've done is they've inserted catalase into the mitochondria where it can scavenge the hydrogen peroxide. Now, there are other hydrogen peroxide scavengers inside the mitochondria, namely peroxyredoxin 3. But this targeted overexpression actually increased catalase over 50 times inside the heart mitochondria. So you can imagine how, with this much catalase, essentially little or no hydrogen peroxide would have a chance of being able to be released from the mitochondria. And when they did the lifespan of these mice, they found that in fact, the overexpression of MCAT, as they called it, led to a significant extension in lifespan. So about 17 to 21% increase in lifespan. So I think this actually to date remains as probably, you know, the most the strongest evidence that altering oxidative stress can potentially have an effect on lifespan. So both mean and maximum lifespan were affected here. And they also looked at a number of phenotypes. Cardiac pathology and cataracts were delayed. Definitely a reduction in hydrogen peroxide production, as you would expect. Oxidative damage to a number of cellular components was down. Aconitase activity, 8-oxo-DG was reduced. And the development of mitochondrial deletions was also reduced. So, you know, this brings up the question. So is this support for the oxidative stress free radical theory and the importance of mitochondria? I mean, it's still, these results are very strong, and now many other phenotypes have been shown to be, or a number of other phenotypes have been shown to be affected in a beneficial way by this increased expression of catalase. So if you put all of these together, you know, we showed that in the SOD2 knockout, the 50% reduction was not enough to affect lifespan. But yet increased mitochondrial DNA deletions did have a reducing effect on lifespan, and mitochondrial targeting of catalase is protected. So with that, I want to just add one more short story to this line of evidence, and that is a story about the naked mole rats. So these naked mole rats are a very long-lived rodent. You can see the picture of them there. They typically live underground. They live more than 28 years, but yet they're about the size of a small rat. So how do they live so long? Actually, we still don't know that. But the interesting part is, even though the fact that they're exceptionally long-lived, they also have very high levels of oxidative damage. And this is just normal. They walk around with this very high level of oxidative damage, and they don't seem to be bothered by it at all. So in fact, if you look at their liver, 8-oxo-DG, the DNA oxidative damage in naked mole rats versus mice is elevated several fold. Lipid peroxidation in a number of tissues is much higher. Protein oxidation, you can see here clearly from protein carbonyl data that it also is much higher in the naked mole rats. So this, you know, really sort of adds another question to our quest to try to figure out whether oxidative stress and aging are linked. So, you know, if we put all of these together, what do they tell us about oxidative stress in mitochondria? So with the role of oxidative stress in mitochondria and aging. So I think, you know, I think, as I said earlier, oxidative stress and mitochondrial function are very important, right? They're important for a number of different cellular pathways. And I think that, you know, perhaps the way the simple form of the theory that Denham-Harman proposed, and that most of these mouse model studies have tested, is probably not the whole story, right? So there's a lot more complexity to the system, and I think more that we need to learn. But certainly it's not as simple as elevated oxidative stress or elevated mitochondrial dysfunction equals, you know, a decrease in lifespan. It just isn't that simple. So, you know, that kind of brings us So the other part of the story, so, as I said, mitochondrial function is important, generation of oxidative stress is important. And I think everyone would agree that this oxidative stress, while it may not be the major player in aging per se, whatever that thing is that causes aging to occur, it certainly is involved in a number of age-related diseases, including cancer, neurodegenerative diseases, and other age-related diseases. For example, my laboratory is very interested in sarcopenia, and we've shown a very strong effect of oxidative stress in initiation and progression of age-related loss of muscle mass and function. So it's definitely still important, even if it isn't part of the basic underlying mechanism. And so with that, I'll end my presentation today, and I hope that this has been a meaningful experience for you. Thank you.